Okay, so good morning, everyone. You are able to hear me, right, everyone?
Yes, sir.
Okay.
Yes, sir.
So, like we had a decision earlier that mainly stressing upon the important topics, we will have some extra classes where we will be discussing all this. Now, genetics, we are continuing in the class. Let us consider that as a separate topic, which we will be continuing in the class itself. And biotechnology is again another topic. We will discuss it separately in our these special classes. If there are any doubts you want to clarify, maybe which you are not able to discuss in the online classes, then we can always discuss them in the school. That is not a problem. But otherwise, as much as possible, try to clear your understanding about biotechnology and all the doubts. You try to clear it in the online class itself. I will be also sharing with you certain materials. Now, what happens is, understand that biotechnology is again a very vast topic. You will see that if there are 2-3 different teachers teaching you biotechnology, everyone will teach you some of the other extra things. Okay, that is because the scope of biotechnology is huge. There are so many things in the biotechnology. There are so many subfields in the biotechnology, right? In a single university, you will see there are different departments who are all working in biotechnology itself. You might be aware that there is a complete now department under DST, that is Department of Science and Technology under Government of India. They have a separate department altogether which is known as Department of Biotechnology, right? DBT they call it as. So all this makes us understand that biotechnology is a very huge subject. It's not only biology, biotechnology, you can say it has biology, obviously, that is the life science is the main thing that you have here. But with that, you will also find other sciences like chemistry is always there, even physics got involved here. You will say mathematics here, you will also see computer science here, right? Statistics you will see in the biotechnology. Okay, so with all these amalgamation of all these different subjects, this biotechnology has started, right? So all the specialized fields we talk about, whether you talk about medicine, even engineering is here in biotechnology. You might be aware that in engineering colleges you have a degree which is now BTech in biotechnology, right? Then there is a field which is called as bioengineering as such specifically. So even mechanical engineering has got into biotechnology now, correct? So there is a huge scope of information. I'm not talking about career. Career comes much later. Before you start thinking about career, the first thing you need to understand is what is biotechnology and what does it contain. So when we say the scope of information in biotechnology, it is huge. It is unbelievably huge. Every day there are new things that is being added to biotechnology. At the same time, if you go into deeper understanding of biotechnology, you will see that it is not an entirely new topic. Biotechnology is almost as old as 8,000 years. Okay, so I will tell you how it becomes such an old subject. Okay, now this syllabus, when we talk about, okay, in this syllabus you do not have so many things as such, but there are some very basic things which are discussed in our class 12 syllabus, very basic things which makes you understand that as such in a very root level what exactly is biotechnology and what all things can be done, what are the scope of this particular field, right? like you are currently studying about genetics, like I told you in the genetics introductory class, biotechnology is a part of what we call as modern genetics, or it's basically the application of our understanding of genetics. Once we understood about the DNA, genes, and once we understood about how the genes are responsible for expression of some specific character or expression of some specific protein, then scientists have started working on how you can manipulate. The main important word in biotechnology is manipulation. Okay. The manipulation word is often used in negative sense, but not always. Even your engineering, when we say engineering, is basically nothing but a manipulation, right? So in biotechnology, you are not inventing something, right? You are discovering something, because it is all dependent upon which is already present in the nature, right? Already something is there in the nature, so those naturally available products, you will carry out manipulation, but with a particular aim, right? The aim is what? When you do this manipulation, you want to develop something which is beneficial for human beings. That is something which need to be fulfilled always. Okay. Because that is why in biotechnology there are big issues which are called as ethical issues. There is one group of people who will always try to say against all the biotechnology processes. Why? Their logic is what you are doing is against the nature, and that is very true. In biotechnology, what we are doing is against the nature, because you are manipulating what is there in the nature. But you have to be able to justify that what you are doing against the nature, it will definitely affect the environment, it will definitely affect your surroundings. But compared to the extent of effect that it will have on the environment, compared to that, you will get more benefits for human beings. That is why biotechnology is not something very easy to do. Okay, if you are found doing something related to biotechnology without a proper approval from certain organizations, there are organizations formulated by Government of India and associated other institutes who have the authority to permit you to carry out a biotechnology research. If you are carrying out some biotechnology activity or research without proper approval from these regulatory authorities, then it will not be allowed. You might have to pay a hefty fee for that, right? So that's why ethical issues are something very, very important. Okay, and that is why we say that this manipulation must be something for the benefit of the human beings. Okay. So this is in brief, and your chapter basically deals with the understanding of the branch of science which is called as genetic engineering. Because we are talking about manipulation, this manipulation, in other word, we can say is engineering. Engineering is also nothing but manipulation. You have something and you then break it down and remake something which is better than the previous one, right? So same here, we have a genetic constitution, but to improve that genetic constitution or to remove something negative in this genetic constitution, we do an engineering of this genetic constitution and try to find or develop something which is improved, something which is better for human beings. Correct? So that is what we are going to discuss here, along with certain applications and all. Right? So if you see the book, then you will get two units here. One is biotechnology principles and processes, right? And the other is biotechnology and its application. Okay, there are applications, but before talking about applications, some very basic principles and processes we are going to discuss. This is where we are going to discuss mainly about what we talk about genetic engineering. Okay. understand. So in the first chapter, we will be talking about the main topics, that is principles of biotechnology, what is being carried out in biotechnology. Okay, so that part, the introductory part, I will not go into too much details right now, because once you understand rest of the chapter, the introductory topic you will understand by yourself. Mainly we will be looking at what are the various tools, because we are saying you are doing manipulation. So when you do some manipulation, like any kind of engineering work you want to do, what do you require? You need certain tools, right? So in genetic engineering, in biotechnology also, you need certain tools. So those tools we are going to discuss. And after that, we will be discussing what are the processes carried out. Okay, what type of processes? In the form of a flow chart, we will discuss one after the other, stepwise, step 1, step 2, step 3, and so on. So that's why, like I told you before, in the class, I believe I have told you once already, that this chapter is not theory. This chapter is actually practical, right? But we are not learning it by doing, we are learning it by reading actually. Practical, normally in a lab, you do it and learn. But here, because our laboratory facility does not allow all these processes to be carried out, okay, does not facilitate so much. So what we will do here is we will learn about these processes, but in a theoretical manner. So that's why you must ensure that if you are attending today's class, then you should not miss any of the classes in future, because this is a continuous process that we are going to discuss. If you miss one class in between, the next class you will not be able to understand, because these are steps. You miss 2-3 steps in between, you will not be able to get the continuity in the next steps, right? So that is why today, whatever, I did not expect so many, but almost 32 students are here in the class, and that's a good thing to know that you people are interested in learning the things extra also. So these 32 students, I would expect them to be present in all the classes. Next class, if there are some new students joining, they will definitely find little difficult, because today's introduction they will not get. And if you do not join in the next class, then after that, in the next classes, you will not find it easy to understand. Okay, so that's why understand that these are practical things we are going to discuss, and you must not miss the class. Now, if we look at the book itself, okay, let us say, like I told you, the biotechnology is not a new subject as such. We often, in some books, although in our book it is not mentioned, you will see that biotechnology is often classified into two types. We call it as classical biotechnology. Okay, these include informations or the processes, products which was developed very long time back, and the other is modern biotechnology, right? To understand what is classical and what is modern biotechnology, first of all, understand this first sentence, very important. What is biotechnology? The definition. Okay, so biotechnology is basically the techniques of using live organisms. Okay, live organisms we are using here. You may talk about microorganisms, you talk about plants, you talk about animals, even human beings, etc., or enzymes from these organisms. Okay, you will use the whole of the organism, or you may be using certain enzymes or certain proteins, or not only enzymes, even some molecules from these organisms. Okay, in simple word, we can say some molecules from these organisms. Why? To produce products, okay, to develop some new products, or sometimes to develop some new process which will be useful for human beings. Okay, that is to use organisms or certain molecules from the organisms, which may include enzymes or other proteins or even DNA molecules, etc. Why do we use it? To develop certain novel, new products or processes which can be useful for human being. So, considering this as the most basic and simple example of biotechnology, if you look at classical biotechnology, you can get an example. One of the most common example of classical biotechnology is curd making, right? Can anyone add here why we can consider curd making as classical biotechnology, or how does it fit into this particular definition of biotechnology? Anyone? How does curd making fits into this definition of biotechnology?
Because we use bacteria.
Yeah. Number one, we are using some organisms which are bacteria. Specifically, we talk about lactobacillus. Okay, so that is fulfilling the first part of the definition, use of some organisms. In fact, that bacteria produces certain enzymes, certain acids which are responsible for making the curd. Then coming to second part of the definition, how is it responsible for making some product which is beneficial for human being? Is there any way we understand that curd is better than milk? Anyone? Do you think curd is better than milk? See, because yes.
It is better for dogs. I don't know about humans.
Why is it better for dogs?
It is less fatty, I guess.
No, not like that. Often curd has some amount of fats. Okay. Normally, you know that milk contain certain proteins which are easy for the small children to digest, which is called as casein protein. And small children, they release enzyme known as renin, which is responsible for breakdown of casein protein or digestion of casein protein. Now, this, if you study the digestion chapter in class 11, you will get it there. This renin enzyme is not produced in large quantity or almost not produced in the adults. And that's why in adults, usually they find this protein to be broken down difficult. Okay. But during the making of curd, this protein is coagulated. This protein is broken down, which makes the curd easy to digest compared to milk. Number one. Number two, remember curd is very good because it is rich in vitamins. Okay, it is very rich in vitamins. So even there are studies which suggest that curd contains substances which are good for preventing cancer and all. So in many sense, curd is a product which is comparatively better than the milk. So here we are using an organism, we are using certain enzymes or certain acids or certain molecules from the organism to convert an existing product. Now, milk is not something we are making artificially, it's a natural product. So a natural product we have manipulated to make something which is better or improved for human beings. So curd is not something that we have started making recently. It is one of the oldest thing, right? Classical biotechnology also includes various types of fermentation processes. Various fermentation processes. One of the oldest product of classical biotechnology, if we say, common is curd, oldest is cheese. There are certain cheese which are almost as old as 8,000 years. I mean, their development, production process, making process has started that long back. Okay, so these all comes under our classical biotechnology, but we did not use the word biotechnology at that time. But we knew that we today understand that these are actually nothing but certain types of biotechnology. Now, when we talk about modern biotechnology, here we usually directly talk about the most important process being carried out, that is genetic engineering, where we are directly manipulating the genetic makeup of an organism. And that's why this particular chapter mainly deals with these processes of genetic engineering, how you manipulate the genes for various. When you go to application chapter, you will see how improved plant varieties are made. Some common examples, if we talk about, there is a plant developed known as BT cotton, where we have introduced a gene from a bacteria into the cotton varieties. As a result, now you do not need any insecticide, pesticide for this particular plant. Why? Because when a organism, when a insect comes and eat upon some tissue of this plant, then automatically it will die, because the plant has started producing some toxic substances for these insects. Okay, so it is called as an insect resistant plant. So you don't need to spray insecticide, pesticide. We all know what are the harmful effects of insecticide, pesticide. Like that, there are different applications found in agriculture: drought resistant plant, flood resistant plant. Okay, then plants which have greater nutritional value, those plants have been developed, right? Did any of you hear about what is called as edible vaccine? Anyone has heard about edible vaccine? No. So if you see vaccine, for vaccine, the worst thing about vaccination process is that it has to be given as an injection. Fortunately, today we have something called as painless injection, but still injection is injection only. Right? Now, scientists are working, very soon we will get such things. Even insulin, we know that insulin is also taken in the form of injections, although some pen devices are used, but still it is an injection. So even for insulin, which is a hormone, and similarly for vaccine, the scientists are developing something which can be taken orally. Okay, when we talk about insulin, scientists are trying to develop some type of tablets or capsules maybe, which can be taken as insulin tablets or insulin capsules, right? So you don't have to take any injection. When we talk about vaccine, scientists are trying to develop certain food products, like they are trying to introduce this a vaccine antigen into a banana. So you take one banana and you get one shot of a particular vaccine. So it's like that. Okay, so these are some of the advances we are looking at. Future, in future we will have it, and in future means not very long, maybe within a few years. Okay, in fact, when we talk about oral insulin is already in the last phase of its clinical trials. Okay, so maybe within few or 1-2 years you will get it in the market. Now these are going to revolution. This will become a revolutionary product as such in the market. Okay, so understand the scope is huge, but yeah, polio vaccine is considered, yes, it is an edible vaccine. You can say polio vaccine is an edible vaccine as of now, right? But as of now, you don't get any other vaccine which can be taken in that form. You can even think further that why it is not possible to take these vaccines in an edible form. So the thing is, these all new things which we are talking about, all these fascinating things which we are talking about, right? We talk about designer babies today. Designer babies is a concept where we talk about having a baby with specific characteristics as per choice, customized characteristics. Right? That is something at a very high level we are talking about. But what we already have today is something called as a gene therapy, wherein if you know that your future child or just born child has some type of genetic disorder, okay, certain such genetic disorders can be rectified by certain techniques of genetic engineering. Okay, so these are all theoretically we are talking about the various scopes. Okay, and soon in future we will have them. Now, so further, there are two things, two definitions given. One is European Federation of Biotechnology has given a definition of biotechnology, which is which encompasses or means which includes both traditional view and the modern molecular biotechnology as such. What does it define? It gives the definition as integration. Okay, that is two things together. There is integration of natural science, okay, and the organisms, or its specific cells only, or the parts thereof, means the parts of the cells, maybe not the entire cell, but only a particular part of the cell. Like I was talking about, certain DNA molecules maybe of the cell. And molecular analogs. Molecular analogs means similar types of molecules. Okay, similar type of molecules which are found in the cells. Okay, which all these things are integrated. It's an integration of all these different things, right? You are integrated all these things. Why? For products and services. Okay, to develop some products or to develop some services. And of course, we should never forget that why we are doing all these things. We are doing all these things for human benefit. So that is our ultimate aim. If you cannot simply do something which is not beneficial for human beings. Okay, so this is your introduction to biotechnology, right? And when we talk about principles of biotechnology, there are two main things we are looking at. That is, we call it as, we say that this modern biotechnology has is born mainly because of two main techniques, core techniques that are possible as on today, which is, one is genetic engineering, means we have learned or we have acquired the ability to alter, okay, to change what? To change the chemistry of genetic material. Means we are talking about the nucleic acid structures. Okay, the nucleic acid structure we are able to change as on today. We have developed such techniques. We have learned how to do this. Okay, so this was one thing which has helped in birth of biotechnology. And the second thing is very, very important. We often do not give much importance. That is maintenance of sterile ambience. Okay, sterile ambience means something which is free of microorganisms, because one thing that is true about microorganism is that they are found to be present everywhere. Everywhere that you think of is has some microorganisms in it. Therefore, to be able to work with only one particular type of microorganism, to work with one particular type of cell or one particular type of tissue, it is very, very difficult. Why? Because there will always be something called as contamination. You say that I want to work with lactobacillus. Okay, easy to say that I want to do some research on lactobacillus. But how will you grow only lactobacillus? You cannot just take a lactobacillus on your table and work on it. Why? Because it will be contaminated by all microorganisms in your surroundings. So what is very important is an ability to maintain a sterile ambience. Sterile ambience means a place, a condition, an environment which does not have any other unnecessary organisms, which is free of other organisms. So there are techniques which we have developed by which we can maintain such a sterile ambience, and thus we have gained the ability to work with specific type of cells, specific type of organisms, and so on. So these are often considered as the two important or the core techniques which has helped in the birth of biotechnology. One is the ability to do manipulation of the genetic molecule, that is the structure of the DNA and RNA. We have learned the technique to manipulate it, cut it, paste it here and there, etc. Literally, there is cut and paste technique which is going on. And the second is ability to maintain a contamination-free environment. Okay, so this was overall the introduction to the chapter.